Hi guys, how are you all? I hope every one of you are fine. I'm your educator Saranya in your YouTube channel GurMantra Shiksha Ka Granth. Welcome to all of you for a session on electron transport chain. Okay, so electron transport chain, when we talk about it, is a continuation from the tricarboxylic acid cycle. We call it as TCA cycle. Also, it is a continuation. Okay, so the products which are formed in TCA cycle are like NADH and FADH2. Those are the electron carriers. While teaching TCA cycle, I told you this important point that NADH, which is produced, in FADH2, these two compounds are nothing but the electron carriers, which has carried electron from the glucose, from glycolysis, from TCA cycle, and these electron carriers are going to give its electrons in the electron transport chain, where the electrons are transported from one molecule to another molecule. Finally, that electron will reach to oxygen, and the oxygen will form water, and all the steps will be completed, which means one glucose molecule will finally be converted into carbon dioxide and water. Okay, so this process actually starts from glycolysis and ends with electron transport chain, which we call it as respiration process. Okay, this takes place in the presence of oxygen only. And also one important thing you have to notice is that electron transport chain happens in the inner mitochondrial membrane. Mitochondrial membrane, okay, inner mitochondrial membrane. Remember this point, note it down because this is very important. This they might ask you in the questions also, okay. So this is very important. So let's start. Electron transport chain is the final step in the aerobic respiration. Okay, so aerobic respiration means it happens in the presence of oxygen. So this is the step. Electron transport chain is the final step where we are going to get ATP. ATP is production happening in the electron transport chain. It is an oxygen dependent process, which means oxygen is involved. It takes place in the presence of oxygen. That's why we also call it as aerobic respiration. Occurs in the inner mitochondrial membrane. The location of this reaction, that is electron transport chain, is the inner mitochondrial membrane. The NADH and FADH2. Okay, so these are the products that is produced in the TCA cycle. So tricarboxylic acid cycle, where NADH and FADH2 are produced, which are the electron carriers. Okay, electron transporters. Okay, they pass through the electron transport chain to give their electrons and reduce oxygen into water. Whenever we say the word reduce or reduction, means hydrogen is passed on to this molecule. Okay, reduction of oxygen means we are giving it hydrogen. So oxygen will be combined with hydrogen to give out water. So two hydrogen will be involved in this process, and a half of oxygen will be involved, so that water molecules will be formed where H2O is produced. So H2 means two hydrogens is there and one oxygen. So here oxygen is having two oxygen is there. So half of oxygen will be used, which means one oxygen and two hydrogens. Okay, so two hydrogens means two electrons which will combine together to form a water molecule. Okay, so reduction of any substance means hydrogen is added to that substance. Okay, that is the meaning. You have to be thorough with these small, important and basic concepts. What is reduction? What is oxidation? All these things. I hope you know this because you've been studying this from, like, I don't know, 8th standard or so. You should be knowing this. So the movement of electrons to various carriers causes the loss of free energy. So this energy is utilized to produce ATP from ADP. Okay. So as the electrons are moving through the carriers, there will be loss of free energy, and these energy will be utilized to produce ATP. Okay. There will be some electrochemical gradient produced when the electrons are transported from one carriers to another carrier molecules. Okay. So electron transport chain or system converts redox energy from oxidation of NADH and FADH2. Okay, so there will be oxidation of NADH and FADH2. I told you the meaning of reduction. Here I will tell you the meaning of oxidation. Oxidation means removal of hydrogen, which means from NADH, NAD plus will be produced, and from FADH2, FAD will be produced. Okay, so oxidation means this hydrogen will be removed. Here the hydrogen will be removed. These are nothing but the electron carriers which carries the electrons. From this, when the oxidation happens, the electrons are released. Okay, and that creates a proton motive force. Okay, this creates a proton motive force where the electrons are transported from these NADH and FADH2 to the oxygen, which is the finer electron acceptor. Oxygen is the finer electron acceptor, final electron acceptor. And since protein motive force is created, that will be useful for the synthesis of ATP. Using the ATP synthase complex in the process of oxidative phosphorylation, which we also call as electron transport chain. Most of the ATP generated during aerobic catabolism of glucose. Catabolism means breakdown of glucose, is not generated from glycolysis. Okay, actually catabolism of glucose, breakdown of glucose is not only taking place in glycolysis, but that continues to TCA cycle and electron transport chain. The amount of ATP produced from glycolysis is very little. Okay, so TCA cycle and ETC, that is electron transport chain, are the major pathways where you will get most of the ATP generated. Okay, that is the process where glucose is completely broken down into carbon dioxide and water. It is derived from a process that begins with moving of electrons through a series of electron transporters that undergoes redox reactions, that is called as electron transport chain. So we have understood the meaning, the definition of electron transport chain. It is nothing but whatever products we have obtained from TCA cycle, that is NADH and FADH2, which are the electron carriers, gets oxidized and electrons are removed from these two products, and that electrons are carried through various electron carriers, and the final electron acceptor is the oxygen molecule, which get reduced to water. Since carbon dioxide has already been released in the TCA cycle, so carbon dioxide and water are the final products in the aerobic respiration, which means glucose is completely oxidized to carbon dioxide and water. Okay, so this is what is happening in the electron transport chain. While the electrons are passing through the various carriers and goes to the water, proton motive force will be created, which will help synthesize the ATP using the ATP synthase complex. That is also we are going to see in the next few slides. Okay. So ATP synthesis, the final product. Okay, because the ATP synthesized in the electron transport chain is very important for our day-to-day activities. Okay, it is very important for our day-to-day activities. Even to move our hands and legs, we need energy that comes in the form of ATP only. Okay, also this reaction happens in the inner mitochondrial membrane. So you have to remember these things like where it happens, what reaction it happens. For example, glycolysis we talk about. Glycolysis happens in the cytosol. Okay, and the next is TCA cycle. TCA cycle happens, where does it happen? Recall it, it happens in the mitochondrial matrix. And electron transport chain, which is the last step in the aerobic respiration, happens in the inner mitochondrial membrane, where electrons are transported through various electron carriers and it finally goes into reducing oxygen to water molecule with the synthesis of ATP. Okay, so this is what happens in the electron transport chain. So in aerobic respiration, the finer electron acceptor is the molecular oxygen. So molecular oxygen is the finer electron acceptor. While in anaerobic respiration, there are other acceptors like sulfate. Anaerobic respirations, you have different electron acceptors that we are not going to discuss in this lecture. So the electron flow takes place in 4 large protein complexes. So in the electron transport chain, you have 4 complexes: complex 1, complex 2, complex 3 and complex 4. They are very large 4 protein complexes. In the inner mitochondrial membrane, they are present. Okay, if you have to also know that not only one of these electron transport chain is present in the inner mitochondrial membrane, there are several. There are several of these electron transport chain, which consists of 4 of these protein complexes present in the inner mitochondrial membrane. Okay, several are there. There is not only one mitochondrial process of this electron transport chain happening. This is the mitochondria and this is the inner mitochondrial membrane. and this is the matrix. This is outer mitochondrial membrane. Here you see there will be several of these complexes present on the mitochondrial membrane. Not just one, several will be there. Okay. So the electron flow takes place in four large protein complexes that are embedded in the inner mitochondrial membrane, together called as respiratory chain or electron transport chain. So all these four complexes of protein together we call it as respiratory chain or electron transport chain. There are several of these respiratory chain present in the inner mitochondrial membrane. As the citric acid cycle takes place in the mitochondria, the high energy electrons are also present within the mitochondria. Okay. This is the mitochondrial matrix. Here only Krebs cycle will take place. That is TCA cycle will take place. And as the TCA cycle is producing NADH and FADH2, that is goes to the complexes present over here in the inner mitochondrial membrane. So all these processes are happening nearby, in the vicinity nearby of each other only this is happening. Okay. As a result, the electron transport chain in eukaryotes also takes place in the mitochondria. Okay. Since TCA cycle is happening in the matrix of the mitochondria, electron transport chain also happening in the mitochondria only, but in the inner mitochondrial membrane. So this is the inner mitochondrial membrane. Okay, so they are happening nearby. As the NADH and FADH2 are produced by TCA cycle, that goes to the respiratory chain here and produces water molecule, like electrons are transported. Okay, this you have to remember. Also one more thing, since you have NADH and FADH which is produced from the TCA cycle, just remember these things, that NADH pumps 3 protons across the membrane and makes 3 ATPs, and FADH2 pumps 2 protons across the membranes and makes 2 ATP. This is a very important point. Based on this point, they'll directly ask you the question. Okay, they'll take the statement, put it in a question and directly ask you the question. I'll tell you how this is happening. But remember these statements, okay? NADH is producing 3 protons, pumping out 3 protons and producing 3 ATPs. And FADH2 is pumping out 2 protons and making 2 ATPs, okay? This process of electron transport chain we'll see in detail. But before that, we will have to discuss some of the basic concepts. The mitochondria is a double membraned organelle. You know, outer membrane is there and inner membrane is there. It's a double membrane organelle that consists of an outer membrane and an inner membrane that is folded into a series of ribs called cristae. The inner membrane is folded into several folds to form cristae. Why is this fold happening in the inner membrane? To give it out a large surface area. More folds means surface area is increasing. To increase more surface area, because there are so many processes happening in the inner mitochondrial membrane, and for that a large surface area is required, and for large surface area these foldings are the responsible one. Okay, so this is called as cristae. There are two compartments in the mitochondria, the matrix and the intermembrane space. Matrix is nothing but the in-between area. This is the matrix, and intermembrane space means nothing but the space between the inner membrane and outer membrane is called as intermembrane space, like this area. Okay. The outer membrane is highly permeable to ions. It contains enzymes necessary for the TCA cycle, while the inner membrane is impermeable to various ions and contains uncharged molecules, electron transport chain and ATP synthesizing enzymes. Very clearly it is given in the statement what it is present in the outer membrane and what is present in the inner membrane. Okay. So the number of electron transport chains in the mitochondria membrane on the mitochondria depends on the location and function of the cells. Yes, this is very important point. The electron transport chain, I have told you that it is not only one, but it is more than that present in the inner mitochondrial membrane, and how many it is present on the inner mitochondrial membrane depends upon its location and function. Okay, if the location requires lots and lots of ATP, then the electron transport chain which is present on the inner mitochondrial membrane will be more. And if a location does not require that much of ATP, then the electron transport chain which is present on the inner mitochondrial membrane will be less. Okay, so I'll give you an example here. In the liver mitochondria, there are 10,000 sets of electron transport chain. Okay, in one mitochondria you will have 10,000 electron transport chains. While in heart, mitochondria have 3 times the number of electron transport chain as compared to that of the liver. Okay, so heart will have more compared to the liver, like 3 times more it is having the mitochondria and so much of electron transport chains, because it requires lots and lots of ATP, because our heart is pumping continuously without any break, right? It is pumping continuously, and for that it requires lots and lots of energy in the form of ATP. That's why our heart mitochondria will have so many electron transport chains. Okay. I hope this concept is easy and clear for you, because these are some of the basic things which you have to know. If you do not know, I've taught you that also. Okay, because there are so many different kind of audience we are targeting here, right? Not only the people who know all the concepts well, but to people who are also weak in their basics. Okay, there are people who are weak in basics, so we are covering them also. That's why I'm teaching you everything in detail from the basics, so that if you have missed the basics in your, I don't know, UG or from 12th standard, you can cover it now. You can understand those basics now and move on with the basics to building the concepts where you can understand everything very well. That is the aim. That is the hope of this education process, right? So this is the electron transport chain where you have so many electron carriers. Most importantly, this is made up of 4 complex: complex 1, complex 2, complex 3 and complex 4. Complex 1 is called as NADH dehydrogenase. Okay, complex 1 is called as NADH dehydrogenase. NADH dehydrogenase, or it can also be called as NADH reductase. Okay, it is called as NADH dehydrogenase or NADH reductase. So it is a proton pump. What happens here is, I'll tell you what happens here. Here NADH is there, right? NADH, that will be oxidized. Oxidation means what happens? Electrons will be removed. That is, hydrogen will be removed. This NADH will be converted to NAD plus, and this NAD plus will be recycled back to our glycolysis and PCA cycle, where they will be used. We studied there that NADH there will be used to form NAD will be used to form NADH. Okay, so this will be recycled back to tricarboxylic acid cycle pathway and glycolysis. Okay, so here NADH will be oxidized, which means electrons will be removed. Electrons will be removed from NADH. See, this blue line will tell you where the electrons are going. Okay, electrons are going to Q. Q is nothing but, complex Q is nothing but ubiquinone. Remember, you can write it down. Q is nothing but ubiquinone. Complex Q is nothing but ubiquinone. I am repeating it again and again so that you can note it down or remember it well. Okay. So this is complex 1. We call it as NADH dehydrogenase or NADH reductase, which is a proton pump also, where NADH here present, you can see NADH will be oxidized to form NAD plus with the removal of electrons. Electrons from here is moving to the electron carrier, that is coenzyme Q. We also call it as ubiquinone. Okay, as the electrons are moving from one carrier to another carrier, electrochemical gradient will be created here that will make the complex 1 pump 4 electrons from the matrix to the intermembrane space. This is the matrix, this is the intermembrane space, this is the outer membrane, this is the inner membrane where your electron transport chain is present. Okay, so 4 electron is pumped by the complex 1. I hope complex 1 is clear to all of you. So what did I tell you? Complex 1 is also called as NADH dehydrogenase or NADH reductase. It is a proton pump. Okay. So electrons, NADH is oxidized to NAD plus, which is recycled back to PCA cycle and glycolysis. Here electron is released. That electrons goes to the coenzyme Q, which we also call it as ubiquinone. In this process, due to electrochemical gradient, 4 hydrogens, that is 4 H plus ions, protons are pumped out from the matrix to the intermembrane space. I hope this complex 1 is clear to you. So let's move on to the complex 2. Complex 2 is called as succinate reductase. Complex 2 is called as succinate reductase. In this complex, FADH2, which is formed right from the TCA cycle, FADH2, that will be oxidized to FAD. And the electrons here again will be going to coenzyme Q, which we call it as ubiquinone. Also, you can see the TCA cycle. You have one step where the succinate is converted to fumarate. For that step, you have an enzyme called as succinate dehydrogenase. That enzyme is provided by complex 2. Okay, do you understand what I'm saying? I'm saying that complex 2 is called as succinate reductase. Okay. The TCA cycle one step is there. Succinate is converted to fumarate, and for that the enzyme succinate dehydrogenase is required. That succinate dehydrogenase enzyme is provided by complex 2. And in this process, FAD will be converted to FADH2. Okay, that FADH2 here in the complex 2 is oxidized. Okay, in the complex 2, FADH2 is oxidized and 2 electrons from that is obtained and it goes to the Q, coenzyme Q. From NADH and FADH2 here, the electrons are transformed or transported to coenzyme Q, which we also call it as ubiquinone. And the second complex is not a proton pump, so from here the protons are not pumped out. Okay, I hope you have understood up to this. Next we are going to complex 3. Complex 3 we also call it as cytochrome C reductase or cytochrome C oxidoreductase. Note it down. The complex 3 we also call it as cytochrome C reductase or cytochrome C oxidoreductase. Okay, in this from Q you can see here Q is there. Q is nothing but ubiquinone. From this complex the electrons will be transported to cytochrome C. This we will study in detail how it is going and all, but now for now you know this. The flow of electrons is from Q to cytochrome C it goes, and as the electrons are transported from Q to cytochrome C, there will be electrochemical gradient created here and 4 protons will be pumped out through complex 3. Okay, so complex 3 is called as cytochrome C reductase or cytochrome C oxidoreductase, and it is a proton pump. Here the electrons are transferred from ubiquinone to cytochrome C. Okay, and through that process 4 protons are pumped out from the matrix to the intermembrane space. I hope this is clear to you. Now we are going to talk about is complex 4. Complex 4 we also called as cytochrome oxidase. It is called as cytochrome oxidase. Here the electrons are transferred to oxygen. Reduction of oxygen is taking place, which means oxygen is getting 2 hydrogens and it is getting converted to water. So oxygen is the final electron acceptor in the electron transport chain. It is starting from the first complex and the electrons are transported to Q, then to the cytochrome C, and finally the electron is accepted into oxygen where the oxygen is reduced to water, and that is the byproduct. Okay, in this process 2 hydrogen is pumped out. As the hydrogens are pumped out from the complex 1, complex 3 and complex 4, the proton gradient is created. Proton motive force is created in the intermembrane space because so many positive ions are here. It is becoming more acidic. So what happens in order to make it equilibrium or to make it a proper process from the intermembrane space, these protons are pumped back into the matrix. From the intermembrane space through the ATP synthase, the protons are pumped back to the matrix. From intermembrane space, the protons are pumped back into the matrix using the F0, F1 ATPase, which we also call as ATP synthase. As the protons are getting pumped back into the matrix, what you will get is you will get ATP. ATP will be produced. Your ATP will be produced as a result. So what is happening in this process? You see, since protons are pumped from the matrix to the intermembrane space through the electrochemical gradient which is happening because electrons are transferred from the one electron to the another electron carrier, and finally it goes to the oxygen where the oxygen is reduced to form water. This proton which is present in the intermembrane space is causing so much acid will be so much there. In order to maintain the equilibrium from the intermembrane space, the proton will be pumped back into the matrix through the ATP synthase or F0 F1 ATPase. As the protons are pumped back into the matrix, your ATP will be produced. This is the actual process of electron transport chain. I hope you understood the flow of the electrons and the proton pump, okay, here, and the ATP synthesis. This is what is happening in the electron transport chain. We'll also see this in detail now. Okay, so I'm going to tell you the name of the complex and what is happening. So the complex 1, okay, complex 1 we also call it as NADH dehydrogenase. So it contains 46 proteins. It contains 46 proteins, and the prosthetic group it has is FMN and iron sulfur centers. Okay, FMN is nothing but flavin mononucleotide. What is this prosthetic group means? They are nothing but the electron carriers. From NADH, the electrons are released, and that electrons is carried to FMN. From FMN it goes to FES. From FES it goes to the Q, the complex Q. Complex Q is nothing but ubiquinone. Okay. So the second complex is called as succinate CoQ reductase or succinate reductase. Here also the number of proteins is 5, and from here the electrons which is produced by FADH2 goes to the complex Q, which we call it as complex Q, which we call it as ubiquinone. Okay. Next is complex 3. Complex 3 is also called as cytochrome C reductase or CoQ cytochrome C reductase. This is made up of 11 proteins, and here the electrons are transferred from the ubiquinone to cytochrome B, H, Bl, cytochrome C1, Fes. Like that, there are several electron carriers in between. It goes to various electron carriers, like from Q, from the ubiquinone to cytochrome B, from that it will go to cytochrome C, from that it will go to ferrous sulfate complex, like that will go. Next is complex 4, which we also call it as cytochrome oxidase. That consists of 13 protons. Proteins. I don't know why I'm saying proteins as protons. Okay, so it consists of 13 proteins, and from this ferrous center, it goes to cytochrome A, then to cytochrome A3, then to CUA, then to CUB. Okay, so these are nothing but the protein carriers which is coming under these complexes. Okay, so complex 1 has carriers FMN and FES. Complex 2 has carriers cytochrome B and FES. Complex 3 has the electron carriers cytochrome B and cytochrome C and FES. And complex 4 has cytochrome A, cytochrome A3, copper A, copper B. Okay, so these are nothing but the electron carriers. Also, we are going to deal with these complexes in detail. So, complex 1 is called as NADH oxidoreductase, or we can also call it as NADH dehydrogenase or NADH reductase. All are the same thing. Okay, it has the enzymes like iron, sulfur and FMN. From NADH, okay, NADH is oxidized so that it gets converted to NAD plus, and 2 electrons are released. That electron is taken up by FMN and FES. Okay. So, and then it goes to coenzyme Q. So, complex 2 is succinate Q reductase, where FADH2 is oxidized to form FAD, and 2 electrons will be released, and the released electron will go to the coenzyme Q, which we also call it as ubiquinone. Okay. Next is complex 3, which we call it as cytochrome C reductase. Okay, cytochrome C reductase. What will happen here is the electrons from the complex Q, that is ubiquinone, will be transported to FES. We saw it will be having cytochrome B complex, then cytochrome C complex. Through all these complexes, electron will be transported. Transportation of electrons will take place. Then it will go to the complex 4, which we call it as cytochrome C oxidase, the fourth complex. From here, the electrons will be transported to cytochrome C, from that to cytochrome A, from that to cytochrome A3. Okay, so all these things are the electron transporters. Okay, then it will go to the copper and iron sulfur complex, and finally the final electron acceptor is nothing but the oxygen molecule that will gain electrons, get reduced to water molecule. Okay, I'll tell you the I'll tell you the process of electron transport chain again so that you can keep it clear. So this is the cytoplasm. This is the mitochondria, outer membrane of mitochondria. This is inner membrane of mitochondria. In between the space which is called as intermembrane space. The space between outer membrane and inner membrane is called as intermembrane space. Okay. In the inner membrane you see there will be electron transport chain. This is the complexes present over here are called as electron transport chain, and this is the mitochondrial matrix. Okay, here in the mitochondrial matrix that the TCA cycle, tricarboxylic acid cycle, takes place. As the product of tricarboxylic acid cycle, you will have NADH and FADH2. They are nothing but the electron carriers. The NADH will go to the complex 1 where it will get oxidized to NAD plus and 2 electrons will be released. This NAD plus will be recycled back to the TCA cycle. This electron which is released from the complex into the complex 1 goes to FMN, FES, then it will go to the coenzyme Q. Okay, FADH2 goes to the complex 2. Okay, as the electrons are produced and it is going into the complex 1, electrochemical gradient will be created that will pump 4 protons from the matrix to the intermembrane space. Remember this. Okay, now FADH2 is going to the complex 2. What happens is FADH2 is oxidized to FAD. In the process, 2 electrons will be released. That electrons will be going to the complex 2. Also remember, if this complex 2 is not a proton pump like complex 1. Okay, after the electrons from NADH and FADH2 goes to the coenzyme Q, which is nothing but ubiquinone. This electrons will be transported to the complex 3. Okay, where it will go to the cytochrome C. Complex 3, as the electrons move to the cytochrome C, what happens here? The protons will also pumped from here due to the electrochemical gradient. 4 H plus will be pumped out into the intermembrane space from the mitochondrial matrix. Now the electron from cytochrome C goes to the complex 4 where final electron acceptor oxygen takes up the electron and get reduced to water molecule. Okay, it gets reduced to water molecule, and in this process due to the electrochemical gradient the complex 4 will pump 2 H plus ions outside from the matrix to the intermembrane space. 2 H plus ions will be pumped outside. Okay, since there is so many H plus ions pumped outside, it has disrupted the equilibrium. In order to maintain the equilibrium, what will happen? The next complex which we have nearby is ATP synthase, which we can also call as F0 F1 ATPase. It will pump the H plus back into the mitochondrial matrix. The H plus which is more in the intermembrane space will be pumped back into the mitochondrial matrix, and in the process of pumping back of mitochondrial matrix the H plus ions, ATP will be synthesized. The ATP will be synthesized as the H plus ions are moving back to the mitochondrial matrix from the intermembrane space. I hope I have repeated the electron transport chain more than 3 times. This should be clear to you by now. From complex 1, the electrons are carried to the complex 4 where it is getting it is reducing the oxygen to form water, and since there are so many H plus ions in the intermembrane space, that is pumped back into the mitochondrial matrix, and in that process ATP will be produced. Okay, in that process ATP will be produced by F0 F1 ATPase. Okay, this is also another diagram which is saying the same things. This is also another diagram which is mentioning the same things. 4 H plus by complex 1, complex 2 nothing, 4 H plus by complex 3 and 2 H plus by complex 4 is pumped into the intermembrane space from the matrix, which is again pumped back to the matrix by using F0 F1 ATPase, where ATP will be synthesized. Okay. Now we are going to see some of the inhibitors which are involved in the process of electron transport chain. Very, very important concept, because this inhibitors they'll ask you in the exam. They'll ask you which inhibitor is going to inhibit the complex 1. You have to say rotenone. Rotenone inhibits complex 1. Malonate inhibits complex 2. Antimycin A inhibits complex 3. Azide inhibits complex 4, and oligomycin inhibits complex, that is F0 F1 ATPase ATP synthase complex. Okay, you have to remember this, very, very important, very, very important. I have everything is summarized in this diagram. Okay, this picture is very beautiful picture which summarizes your electron transport chain, like how it is happening. Also it is telling you like what are the complexes present here, how is the electron transported, finally how it is reducing oxygen, and how water molecule, it is not H2O2, it is H2O. Okay, how oxygen is forming here, I mean water molecule is forming here, and about the complex one which is called as NADH dehydrogenase, which is composed of 47 subunits also. And what is the compound that is inhibiting the complex one is rotenone. Please note it down. This is my sincere request, very, very important slide. This is very, very important slide. They will ask you so many questions from this, and every answer for those questions is present in this slide. So the complex 1 is inhibited by rotenone, complex 2 is inhibited by malonate, complex 3 is inhibited by antimycin A, complex 4 is inhibited by azide, complex 5 is inhibited by oligomycin. So remember, rotenone, malonate, antimycin A, azide and oligomycin. Okay, see, these are very important inhibitors, you have to remember it. Okay, you have to remember it, practice, write and see, you will remember it very easily. Okay, so electron transport chain products, from glycolysis you are getting 2 ATP. Okay, that is the ultimate yield, and 2 NADH you are getting, that will produce 5 ATP. Next, from pyruvate oxidation, that is the link reaction, you are getting 2 NADH, that will also give you 5 ATP. From TCS cycle you are getting two, actually you are getting GTP, but you will consider it as ATP. So 2 ATP you are getting, 6 NADH which will give you 15 ATP, 2 FADH2 which will give you 3 ATP. So totally from all of this NADH and FADH2 are not as ATP in the TCS cycle. To make it into ATP, like electron transport chain is used, so that this NADH and FADH2 transport its electrons through the electron transport chain and make ATP using F0 F1 ATPase. So finally in the electron transport chain you are getting the ATP made. So in the aerobic respiration, if they ask you how much I mean ATP is made in the aerobic respiration, you have to say 32 ATP. 30 to 34 depends. It depends, but you can say 32 ATP will be made. Okay. From glycolysis, 2 ATP will be made. From PCA cycle, only one. I mean, if it is happening 2 times based on 2 pyruvate, 2 ATP will be produced. And this NADH and all will be produced, but that will be converted to ATP in the electron transport chain. So finally, in the process of aerobic respiration, you will get 32 ATP. This chart is very important. Okay, I've taught you about glycolysis, pyruvate oxidation, that is the link reaction, and citric acid. Now I've taught you the electron transport chain also. Very, very important concept. So many questions they'll ask you from here. NADH generate more ATP than FADH2. For every NADH molecule that is oxidized, 10 H plus ions are pumped into the intermembrane space. This will yield about 3 ATP. Okay, remember this, 10 H plus will give you 3 ATP. Because FADH2 enters the chain at a later stage in the complex 2, only 6 H plus ions are transferred to the intermembrane space. This accounts for about 2 ATP molecule. Actually, FADH2 will be going to the complex 2. That will not pump any protons outside. They are talking about the next complex. Okay, so that will produce 2 ATP. Okay, here. So totally 32 ATP molecules are generated. Okay, so 32 ATP molecules are generated in the process of respiration. Okay, So I hope you understood the electron transport chain very well, because I've repeated it more than 3 times with the diagrams where you have how the electrons are transported from the NADH, FADH to the final oxygen, which is the electron acceptor, which is getting reduced into water molecule, and how due to the electron transport, electrochemical gradient will be produced, and protons will be pumped out into the intermembrane space, and that is disrupting the equilibrium. Since so many electrons are pumped out, proton motor force will be created, and that protons which are pumped into the intermembrane space has to be pumped back again into the mitochondrial matrix through F0 F1 ATPase or ATP synthase, and in that process ATP will be produced. This is the concept, this is the story of your electron transport chain. Comment below on how did you like about this electron transport chain. Like, share and subscribe to our YouTube channel GurMantra. I hope you understood this concept of electron transport chain. Keep revising it, keep practicing. Okay, so that is our mantra. What is our mantra? Study, revise, practice and repeat. Thank you so much. Have a nice day.